Good day, everyone. So today we will be having an in-depth investigation into a little bacterium that causes all kinds of havoc, and we will discuss this shortly. So what's the situation? During an extended period of rainwater, a sheep farmer observes patchy hair loss and inflammation of the skin in several sheep. His veterinarian takes samples, and after receiving the laboratory reports, he makes the diagnosis of dermatophilosis. So, which little organism is actually causing so much havoc? This organism is Dermatophilosis congolensis. And you might be wondering, what is this exactly? And this little organism is in fact a bacterium.
The bacterium is of the order Actinomyces. It was previously believed to be a fungus because of the holocarpic form. The bacterium forms septa across the planes and then fragments. The fragments form Sarkini-like packets and then develops into separate corky. A bacterial nucleoid can be seen in the electron micrograph and multiple ribosomes scattered across the cytoplasm. The penetration into the victim's skin is limited to the extent to which nutrients can across the hypa in nutritious regions in the skin, and the availability of CO2, as both the factors are important for the organism's growth and development across the skin. The bacterium has a hypa that exerts a mechanical strength that aids in the penetration into the skin. The protecting capsule covers all the stages of the bacterium except for the free zoospores. The capsule consists of a polysaccharide gel with a surface that is hydrophobic, causing it to be sticky. The bacterium is gram-positive and is non-acid fast. The bacterial infection spread by means of three ways, namely direct contact with animals that are infected or animals that carry the bacterium, through mites, contact with the contaminated surroundings. Spreading can also occur by means of vectors like ticks and flies that transport the bacterium.
Do weather conditions play a role in outbreak? Prolonged wetting by rain, high humidity conditions, and high temperatures. Can it survive outside or inside the host? It can survive outside of the host and is naturally occurring in the soil, though currently not yet being isolated from the soil in pure culture. It can also survive inside the host as it lives inside lesions on the skin.
Which is the most common portal of entry, through direct contact of areas with low protective covering on epidermis. Which specific mechanism facilitates the exit route? Zoospores are released from lesions.
Given the characteristics of this organism, what kind of samples would you take to send to the lab? Scrapings of the crusts on the skin may be used. How will you preserve and transport these samples if required? Zoospores may be transported as the scrapings. Filamentous hyphae may be transported in sterile saline solution in order to prevent desiccation. Indicate how this organism differs from other bacteria, viruses, and fungi with special reference to classification, morphological characteristics, reproduction, genetic exchange, and lab identification techniques. For this question, we will be dividing it into 4 parts and looking at each of them individually. Dermatophilis congolensis. In terms of classification, it is a bacterium of the order Actinomyces. Under morphological characteristics, it is dimorphic due to the fact that it has filamentous hyphae and motile zoospores. The filaments fragment into cocci, and it is a facultative anaerobe. In terms of reproduction, it undergoes binary fission, produces zoospores, and can reproduce using conidia, which are developed in chains from the aerial hyphae. For lab identification techniques, it is a gram-positive bacteria, but the complex cell wall makes this test unsuitable. Therefore, fast Giemsa or Romanowski stains can be used in fresh cuts. Other bacteria. In terms of classification, they are prokaryotes. In morphological characteristics, they may be rod-shaped, cockeye-shaped, or spirochete-shaped. They may also be pleomorphic, in which case they may assume many shapes. In terms of reproduction, they undergo binary fission. In terms of genetic exchange, there is mutation, integration, recombination, conjugation, transduction, and transformation. For lab identification techniques, a gram-positive test may be used.
Looking at viruses, the viruses are classified as just microorganisms. The morphological characteristics of viruses, they have a nucleic acid core, outer protein which is a capsid. They can be enveloped or naked. They are acellular, means that they are lacking cytoplasm, chromosomes, and other organelles. In terms of reproduction, they replicate using host cell machinery. Genetic exchange of viruses is via mutation and recombination through co-infecting host cells. How to identify through lab techniques is only done through electron microscopy. Looking at fungi, they are classified as eukaryotes. Their morphological characteristics are that they also have hyphae, spore types, but the hyphae are much larger than in the actinomycete. Reproduction is asexual using conidia, sexual using fusion of nuclei. Genetic exchange is through nuclei undergo meiosis and fuse. Lab identification techniques are that they are used lactophenol, cotton blue stain used in wet preparation to visualize structure for microscopy, methylene blue or gram stain for yeast, periodic acid Schiff, and Grocott methenamine silver. And thus concludes our investigation.